this series of videos, we're going to be learning in detail the role of DNA as the repository of the genetic code in all organic life on Earth. Now, when we were first exposed to DNA, we learned that the way that it acts as the repository of the genetic code is that it is the chief regulator molecule in determining which proteins a cell produces and how much of each protein are produced. The problem is that as a storage molecule, DNA does not actually do anything directly, with one exception. The primary function of DNA, rather than to produce protein directly, is to produce the intermediary molecule RNA, which takes a more direct hand in the production of protein. Now, when we learned that RNA is the carrier, or shall we say messenger, of the genetic code, we were actually referring to a specific form of RNA which we see being produced here, which, not surprisingly, is what we call messenger RNA, which can be shortened to mRNA. And when we refer to RNA as being the carrier molecule of the genetic code, it is specifically referring to messenger RNA. So what we observe in this diagram here is the production of mRNA, or messenger RNA, using a procedure that is very, very similar to the process of DNA replication. So we see that there is an enzyme here that is separating the two strands of a DNA double helix, and then each of these single strands of DNA can be used as a template molecule in the same way that single strands are used as a template to copy the whole DNA molecule. The difference is that instead of building a daughter molecule that is made of DNA, this daughter molecule is made of RNA instead. And this RNA molecule, this piece of mRNA that is complementary to a specific sequence of DNA, is what we call a transcript. And therefore, we can conclude that this procedure of opening a DNA strand and copying a complementary strand of mRNA based on a template is what we call transcription. And this is the first half in the process of doing protein synthesis. But obviously, we need to use the mRNA molecule in order to directly make a protein. So when the mRNA transcript is produced, it migrates out of the nucleus and eventually searches for the enzymes within the cell, which are what we call ribosomes, in order to make a protein. And this process of reading an mRNA transcript and translating that into an amino acid sequence that will make up a specific protein, not surprisingly, is what we call translation. And you can remember this term because we are effectively converting between two different molecular families, or languages, if you will, where our template molecule is a nucleic acid mRNA, which is then being translated into a different molecule, or different language, if you will, because proteins are made of amino acids instead of nucleic acids. So the last thing that we're going to talk about in our introduction is reading DNA itself. Now, the reason why we don't necessarily use DNA and the genetic code interchangeably is that 100% of your DNA is not actually used to produce protein. And we'll learn that a lot of DNA actually has regulatory function instead, which is used to determine which specific DNA is transcribed into mRNA and then becomes a protein. And then there is some sequences of DNA that are just used to make RNA, but that's where the function ends. And then there are some sequences of DNA that actually don't have any function at all. And unironically, the technical term for DNA that really doesn't do anything is what we call junk DNA. Now, what we're interested in is the DNA that is ultimately used to produce a protein. So any DNA that can be transcribed, and then of that DNA that is transcribed that ultimately gets translated into a protein, these are what we call genes, and this is why the human genetic code does not actually include 100% of your DNA, because it specifically refers to the DNA that is ultimately used to make protein, and all of the genes that are found within your DNA together are what we call your genome. So the Human Genome Project is specifically interested in looking at the DNA that ultimately decides which proteins your cells are going to produce. Now, as we'll learn in the subsequent videos where we look at transcription and translation directly, gene structure is actually relatively predictable, and we can learn a lot about the structure of DNA based on how we read genes. So we know that DNA molecules have two strands because the default DNA molecule is an anti-parallel double helix. So one will be read from 5' to 3' this way, and the other one will be read 5' to 3' this way in the opposite direction. Nothing new there. Now, based on the process of transcription and translation, we can see that an mRNA molecule is produced using a DNA template that is only found on one side of the DNA strand, or only on one DNA strand, which is why there are two sample genes here, and these genes are both found on the DNA strand on the right. So because genes are only found on one side of your DNA, it is possible to actually find genes on either strand of the DNA, and it is even possible for some of these genes to overlap with one another if they manage to share the same sequence of nitrogenous bases. So it is possible to have a third gene right here, and it is even possible to have another gene right here overlapping with these two. And when we learn the different components of what makes up a gene, we'll see how this is possible. In the next video, we're going to look specifically at the process of transcription and how your enzymes know where a gene begins, where it ends, and what the different parts of a gene ultimately mean in the process of producing an mRNA transcript.